Hello and welcome back to Microbial Concepts. So we are going to discuss about another extremophile here, that is halophiles. So you might have a different topic, okay, for your practicals like studies on halophiles which are isolated from seawater, okay, then the pigment isolation or the pigment production or salt tolerance, etc., etc. Okay, so it might vary from a little bit from this video, but the method of isolation of halophiles then identification is the same. Okay, so I have simply given the title here as isolation of halophiles. Okay, same I did it for acidophiles, then for thermophiles, etc. So it depends from college to college. The title may vary, but this is the isolation protocol is going to be the same. Now about this kind of extremophile that we know, so they love salt. Okay, that's the term halophile. It means that they are salt loving bacteria. So they thrive in environments with high salt concentrations. Okay, so this particular term is derived from a Greek word that means halos, meaning salt, and phylos means loving. Okay, so very simple to remember, halophiles are salt loving bacteria or microbes. Now, halophiles, they can be broadly classified as slight halophiles, moderate halophiles, and extreme halophiles, okay, or hyper halophiles. Now, why this? Because the salt concentration can vary from genus to genus of a given microbe, right? That's the reason why it is necessary to classify them. Now, the slight halophiles, these organisms, they prefer environments that are relatively low salt concentrations. Okay, that means 1 to 3% of salt max, right? Then moderate are little higher than 1 to 3%, that is 3 to 15%, and above 15%, those who are uh, evolved and adapted to extreme salt concentrations, they are categorized here, that is extreme halophiles or hyper halophiles. That means salt concentration is very high, 15 to 30% of salt. Okay, and they can be found in places like Dead Sea or salt flats. Now what are salt flats? If you are not aware what this particular environment is, you can Google it up. Now about mechanisms of salt tolerance, right? How they got adapted, what are the mechanisms that the bacteria it uses to get adapted to a high salt concentration environment. That is very important because you will get this question in your vivas. Okay, so I have listed here three mechanisms, very easy to remember. The first one is very simple and important, that is osmotic balance. Now, halophiles, they maintain osmotic balance by accumulating some compatible solutes. Now, what are the compatible solutes? Example, potassium ions, okay, or organic compounds like glycine, okay, to counteract the high external salt concentration, to balance or to act against the higher external salt concentrations, the bacterial cell will start accumulating these kind of compatible solutes. Okay, they are compatible with, they are okay with accumulating those. Okay, then second is protein adaptation. Now proteins of halophiles, they are adapted to function in high salt concentration. Like I said, these are extreme conditions, right? Now here the extreme condition is the salt concentration is very high. So to function properly, the proteins, the enzymes of the bacterial cell, they also need to adapt to this high salt concentration. So this adaptation is made, and they often have higher content of acidic amino acids which help them remain stable and functional in the presence of high salt concentrations. Okay, now cell membrane adaptations is the third one. The cell membranes of halophiles are highly specialized, and they contain unique lipids that help maintain membrane integrity and functionality in high salt concentrations. Then next are examples of halophiles, because this can be a question in Viva. You should know few examples. So first is Halobacterium salinarum, then Haloacrula marismortui, then Halococcus, Halomonas elongata, and Dunaliella salina. Okay, now you can see first four there is a term halo added. Okay, the prefix is used, that is halo. That means it denotes that they are salt loving bacteria. Okay, then next is now about sampling environments. What are the environments from where you can get a sample as you need to proceed with isolation for your practicals, right? You need a proper sample. So here are a few examples of environments that have high salt concentration. So first is salt flats. That means it is kind of a salt desert. For example, in India, we have Great Run of Kutch, right? Then second is seawater, very easy. Okay, to get the sample. Then you have coastal salt marshes. You need to Google out a nearby area if there are any coastal salt marshes. Then you have saline soils in arid regions. Arid regions are very, very dry regions, okay? As there is no rainfall, the soil, it naturally it has high salt concentrations. Then fifth is agricultural fields with high salinity due to irrigation, okay? So now you have your sample. Okay. Suppose you got your sample. Now you need to proceed with the isolation. So what you need first, you need to know what kind of medium you need to use here. Okay. For isolation, as we are targeting a desired type of microbe, right? So two commonly used media for isolating halophiles are here mentioned here, like halophilic agar HA medium. Now, this medium, it contains high concentration of salt, typically NaCl, right? That is what we are aiming here to create a suitable environment for halophiles. And it usually includes all other nutrients like peptone, yeast extract, trace minerals to support the growth of a wide range of halophilic organisms. Then second is modified growth medium, MGM. This is a versatile medium that can be used, that can be adjusted to different salt concentrations depending on the specific halophiles being targeted. Now, it generally contains a carbon source like glucose, then it has nitrogen, it has vitamins, essential minerals with varying levels of salt, that is NaCl, to cater the specific halophilic requirements. Okay, so that's MGM, modified growth medium. Now, you can use any one of this. If you want a detailed um, media composition, you can Google it out. Now, from research paper to research paper it may vary because few scientists they modify and then they use a particular medium depending on their sample, depending on their aim. So please read that and then you can follow. Then next is about the protocol. So now we have collected our sample, right? We have our sample, we prepare our media, use halophilic agar or MGM, then you inoculate your media with sample. Okay, now here if it is water you do serial dilution, or also you can directly take a aliquot of 10 microliters and check the uh, what amount of microbes are present. Are you getting a crowd plate, or are you getting um, minimum 30 to 35 colonies, isolated colonies already? Then it is okay, you don't need to go for serial dilution. If you are getting a plate, okay, crowded plate, then you need to go for serial dilution. If it is soil, then yes, you have to do serial dilution and then you need to inoculate your media with the sample. Then you incubate that for optimal pH, say 37 degrees Celsius, and you observe the growth for say 2 to 7 days, because this is extreme environment that we are creating here. We are trying to mimic this extreme environment in our lab. So it may happen that some microbes they need time to adapt and grow. Okay, so never report your uh, results in 2 days. Okay, at least try to wait for 5 days, see or try to observe if there are any new colonies or there is any other observation, okay, that you can report. Then once you got the different types of colonies growing on your plate, then you can go for streaking for isolation on fresh media. Now here you can use a plate for master culture, or you can directly go for slants and you can preserve it if you want to work on it later. Again, you need to incubate those. Okay, then you get a pure culture, you can use that for identification, and also you can store it by sticking them on a slant. Now this protocol, it depends on how you want to work, how you want to plan your experiment. Okay, now after getting a isolated pure colony, you go for characterization of your isolated halophile. Now, first thing that we do is reporting the colony characteristics, then going for gram staining motility, and then for biochemical test, right? This is the basic protocol for isolation of halophiles. Now, once you have done isolation, if you have identification in your syllabus for practicals, you go for identification. Otherwise, it is okay for just doing the isolation. Okay. Now, for identification, what you need to do, you need to get a fresh 24 to 48 hours fresh culture, or the fresh colonies, pure colonies, isolated pure colonies. Then you write down their colony characteristics, you perform gram staining, and you start conducting the biochemical test. Okay. Now, depending on your gram staining motility, you refer to the Burgess manual for identification till the genus level, and you plan what kind of biochemical test you need to perform. Okay. And then you can, depending on the results of biochemicals and gram staining and all other tests, you can try to identify till genus level. This is for college level practical. Okay, but if you are doing a research and you need to identify, and you need to confirm 100% sure with your culture that what culture you are working with, you need to do uh, gene sequencing, okay, for identification. So first thing, you extract DNA from your isolate, then you perform PCR amplification of 16S rRNA gene. You sequence the 16S rRNA gene, and then you compare the sequence with database, okay, for example using BLAST. Then you identify the closest match, and you confirm the identification with additional test if required, or if you get match till 95%, 99%, it is okay, you can go with it. Okay. So that was about identification. Now, other than this, other than our practical work, our lab work, you need to know a little bit more about halophiles, because you will get questions in your viva, right? So first is here ecological and industrial importance. Why to study halophiles, right? What is their significant role in our ecosystem? You should know. So first is biogeochemical cycles. Now halophiles, they are crucial in nutrient cycling, particularly in nitrogen and carbon cycles. Then in bioremediation, as they can be used to clean up contaminated sites, okay, or contaminated with pollutants, as many halophiles, they degrade toxic compounds under high salt concentrations as well. Okay. Like oil spill in sea. Okay. So in that, I'm giving one example here. So in that case, which microbes we can target? We can target one that can survive in high salt concentrations, right? So that's the way we think for bioremediation using microbes. So if you are new to this term, you don't know about bioremediation, then you can watch uh, two videos which are already uploaded on my channel, and those are handwritten notes. So I think they might be more useful to you. Okay, as you have bioremediation in theories also for most of these students. Then in third importance, we have biotechnology. So halophiles, they can be exploited in biotechnology for production of enzymes, for then bioplastics and other biochemicals, as they have very stable and functional enzymes which can work under extreme conditions, right? So for instance, haloenzymes, that means halo, again salt uh, tolerant or salt loving microbes which produce enzymes, they are termed as haloenzymes. They can be used in processes that require high salt concentration. Then again, in food industry, we have the importance, as they contribute to the fermentation, preservation of certain foods like salted fish, soy sauce, by producing unique flavors and textures. Now about salt tolerance test. This is extra information here. It is not necessary that you have to do this in your practicals, or you will have this in your practicals. It depends on your syllabus. So if you have this in your syllabus, now suppose you have this, so you need to know what kind of test it is. It is a biochemical test, okay, and it tells you or it gives you more information like till what level or what concentration your microbe is able to tolerate the salt. Okay, that's the meaning of salt tolerance test. So it is a biochemical test used to determine the ability of microbes to tolerate high salt concentration, like say 6.5 to 7 or 8 max. Okay, you can go till 8 for your lab level practical. Now, salinity is an important factor which affects bacterial growth. We know that, right? So, the sodium ions are essential for physiological functions in all organisms, but a certain amount of sodium chloride is necessary for survival. Now, the salt tolerance and required NaCl levels, they may vary among different species. Now that's the reason why we need to do this test when it comes to halophiles. Now bacteria that grow ambiently in salt concentration of 1.5 less than 1.7%, okay, that is around less than zero point 0.2 to zero point 0.3 molar, they are considered as non-halophiles. Okay, so these are our normal bacteria. Okay, we don't classify them in our halophiles. Okay, not slightly, not moderately, not, they are not halophiles only. Okay, so remember that. Now salt concentration of sea water is approximately 3.5. Okay, approx, here and there you get the salt concentration of seawater as 3.5, that is 35 grams of salt per liter. Okay, so you can see how salty our seawater is. So depending or keeping this in your mind, you can plan your salt tolerance test, that how much concentration you need to test. So we have here slight halophiles, moderate halophiles, extreme halophiles, and halotolerant bacteria. Now, what are their NaCl concentration requirements? So, slight halophiles, they require NaCl from 1.7 to 4.8%. Okay, um, it varies from different species. Okay, remember that. Always remember it for all of these. Then moderate halophiles, they grow best at concentration of 4.8 to 20. Extreme halophiles 20% to 30%. Okay, and halotolerant, they can tolerate salt concentration of 2.5 molar without requiring it for growth. That means the salt, that much salt is not required for their growth, but still in that condition they can tolerate the external salt concentration pressure on the cell and they can grow. Okay, so you can see halotolerant bacteria, that is 2.5, so somewhere near moderate halophiles. Okay, so in these kinds of environments you can also find some bacteria that are halotolerant. Okay, now the media used is tryptic soya broth with different salt concentrations or modified salt broth. Now modified salt broth, you can see the composition is given here: HMH peptone, that is hard digest peptone, then peptone, glucose, sodium chloride. It depends now, depending on what concentration you want to test: 4.5, 5.5, 6.5, etc. And bromocresol purple as an indicator. And the pH is around 7.2 plus minus 2 at 25 degrees Celsius. Now, the procedure is using a sterile inoculating loop, pick up a well isolated colony of a sample bacteria which is freshly grown. Okay, or the culture should not be too old. Okay, so max till 24 hours. Then you transfer one or two loopful of bacterial suspension or your fresh broth culture into the given medium. Now incubate these tubes aerobically for 24 hours at 35 degree plus minus 2 degrees Celsius and observe the visible growth, that is turbidity. So uninoculated tube is this one. Okay, you can keep it for the control. This is a negative one, that means the culture cannot grow in the given salt concentration. Okay, and if you see turbidity like this, then yes, the culture is able to grow in given salt concentration. Okay, so what level of salt tolerance your bacterial culture can have, you can determine likewise. Okay, so if no growth is seen, re-incubate the tubes and observe the growth for up to next 72 hours and check for turbidity or color change at 48 to 72 hours. Okay, so this is about. Now moving towards viva questions. So I have listed around 8 viva questions here. Now, very first is you will get what are halophiles. Okay, very basic. So I'm not going to answer that. Now, next is why it is important to use specialized media like halophilic agar for isolating halophiles. Now, we are using this specialized media to provide high salt concentrations, and we are trying to mimic the natural environments, okay, in our lab. So this helps us in isolating halophiles in our lab. Okay, now what is the significance of streaking for isolation when culturing halophiles? Okay, not just halophiles, acidophiles, okay, or thermophiles, etc. Now, streaking helps us separate individual colonies, ensuring that we get a pure isolated culture for our further studies, further test or experiment. Okay, now, how do you determine if a colony which is growing on a halophilic agar is a halophile? Now, the best way to check whether it is a halophile or not is you go directly for salt tolerance test. But also with that, you can go for gram staining and biochemical test. Read your Burgess manual. Try to identify or try to check whether the genus that you are looking for that you have identified is actually a halophile or not. Okay, then what temperature and incubation period are typically used when isolating halophiles? So halophiles, we often incubate them at optimal growth temperatures, that is 35 to 37 degrees Celsius for 2 to 7 days to allow for sufficient growth, and then we report our results. Then next is, can halophiles grow in environment with low salt concentrations? No. Now, no, because halophiles means they love salt concentration, high salt concentrations. Okay, so only slight halophiles that uh, grow within range of 1 to 3%, right? They can grow in low salt concentrations. Okay, and they can tolerate such low salt concentrations, but not the moderate and extreme halophiles. Okay, now, how do you ensure the purity of isolated halophile colony? So by streaking for isolation, by visually inspecting colonies to ensure they exhibit uniform colony morphology and growth characteristics, then you can go for gram staining, okay, to ensure the purity. Then next is, how can molecular techniques like PCR and sequencing they help in the identification of halophile? Now, PCR amplifies specific genetic markers, like for example 16S rRNA gene, which can then be sequenced and compared to the database for species identification. Okay, so yes, nowadays uh, most of the researchers they go directly to the molecular techniques for identification. Okay, this is the reason, and it saves their time and it is more reliable. Now, next is, what are some potential applications of halophiles in biotechnology and industry? Now, potential applications, we have already done this slide, right? So let's just revise them quickly here. So we can use them in various biotechnological processes such as enzyme production, bioremediation. Then we have applications in food preservation and pharmaceutical industries as well. Okay, so these are few viva questions which I have listed here. You can think of more. You search your answer for your questions on internet and get ready for your Viva. Okay, so I hope this video is helpful to you all. Do subscribe to my channel. Do like my videos, share my videos with your friends, and keep learning, keep enjoying. Thank you.